Should we go ahead or give it another minute or so?
I think you can go ahead and get started. Deepsha, if you could start the recording.
Yeah, I started it already. Yeah.
Okay, good. Sorry.
Okay, good morning, everyone. Nice to see you all back. Hope you all had a good evening, good night, good morning. I'm not sure, all different time zones. I hope you're all ready to look at life in 3 dimensions. So today, what I'm going to focus on is how looking at things in 3 dimensions suddenly gives you a completely different picture about what you've been thinking about a protein, and how that visualization in 3 dimensions actually is integral to the function of all of these proteins, right? So again, like yesterday, please feel free to send in your questions, ask your questions anytime. You can interrupt me at any point. Again, it'll be, I hope you all have had your coffee and you all have all the energy to interact, correct? So I'm looking forward to a very interactive session again. And let's get started. So, right. So first, I just want to briefly look at what we did yesterday. Do you all remember anything that came to your mind of what we did yesterday? We actually defined bioinformatics and databases. Do you all remember what the definition or a simple definition of bioinformatics that we finally came up with? Any recollection of what we talked about? Yes, absolutely. Integration of all omic terms. It's fantastic. And then we actually looked at all the key players in the field of bioinformatics. And I'm sure you all remember one figure whom we defined as the father and mother of bioinformatics. Do you remember her name? Any? Fantastic. Wow, you guys are fantastic. So Margaret Ahoff, right? So every day I want you to remember a few things, and hopefully that stays in your mind, right? So that it rings a bell when you are asked about this. So we also talked about sequence explosion, big data, and all of those things, genome sequencing. When did it all start? Why is all different aspects of the genome important? Not that one aspect of the genome is more important than the other. Now we actually have to focus on pretty much the entire genome, right? And why it's important. There are lots of different things. Also, all the challenges we're facing today because of this explosion in data. While we have these challenges, but we also have solutions to these challenges, right? So we discussed a little bit of the solutions to these challenges. Without this explosion of data, of course, it's a good thing, although it's challenging. It's a good thing because it's going to keep us all busy for the next, 20, 30 years or 40 years or whatever may come, right? So we all have something to contribute to, so the explosion. So before I start today's lecture, are there any questions that's lingering in your head from yesterday that you want me to clarify before we get started? If you have any questions, I'll just wait for a second or two to see if you have any questions so that we can actually move forward. Or we can actually, you can keep those questions and we can actually discuss it at the very end of the lecture. Hopefully I'll leave some room for questions. Of course, we also talked about Human Genome Project, HapMap Project, ENCODE Project, Genome-wide Association Studies. Where can we access recorded sessions? I'm sure Deepsha will send you an email. Okay, so role of big data. So when you look at all of these different projects that we talked about, Human Genome Project, HapMap Project, ENCODE, and all of these things, what is this that came out as, if you are to summarize everything that we have learned about all of these projects and the role of these projects, and what is it something that actually is a take home from all of these projects, other than we're drowning in data, right? It's contributed, and it also has contributed to the way we look at diseases. So is there anything that's just coming out of these that you did not know that you were actually shocked to know? starting to map the data. That's correct, Karina. So you actually are beginning to map the data, begin to understand. And also for me, the overwhelming thing, the effectiveness of medicine. That's right. So bettering personalized medicine, pharmacogenomics, that's fantastic, Jeffrey. Yes, that's pretty much what it is, right? So we cannot assume that one drug is going to work for all of us. And you cannot assume that because we are 99.5% or 99.9% similar in our genomes, that we are identical. We are actually not. In fact, even identical twins are not identical, right? So we are learning a lot, but it's a good thing that we're having so much data because we can figure out, it's like a puzzle, right? I'm sure all of you love to put all of these 1,000-piece puzzles and all of those. It's pretty much like a puzzle. We're all trying to figure things out, right? So now I'm going to take you, this is a repetition, right? So today our learning objectives is going to be looking in 3 dimensions. What can structure tell us? How is structure an integral part of, without structure, why is it integral to understanding functions of all of these things? And how one single mutation in a protein sequence can affect its 3-dimensional structure in a way it changes its behavior completely. And as I told you yesterday, any change in a gene, it gets personal. If it gets personal, that means it affects us, right? So which means that's what we call, if it's not normal, then we call it a disease, right? So that's pretty much what we're going to focus on today, right? So introduction to protein structure, right? So I'm sure all of you have seen all of these pretty pictures, right? And I must tell you that a huge amount of work goes behind before we can actually see this pretty picture, right? That's my expertise. That's what I'm trained in. And so I've gone through the pain and suffering of actually determining the 3-dimensional structures of proteins, right? So what is this? This is the first structure of protein that was solved. And that's something won a Nobel Prize as well. Any guesses of what this structure is? Just fill in the blanks. Anything? The whole, yes, you're right. It's alpha helices. Is that what you're meaning? Or just the blue? Yes, but what is the name of this protein? This is where you kind of guess. It starts, I'm going to give you a clue. It starts with the M. Anything that's actually very important protein in our bodies. Absolutely, Chandrani, and Chandrani is correct. It's actually myoglobin, correct. This was the first structure of a protein that was actually solved. And because it was so important, actually it won a Nobel Prize, right? And then it was shared by Max Perutz and John Kendrew in the 60s, long time ago, in chemistry, and it was divided between these two, right? This was the first time, and you can actually imagine the awe that people had when they actually saw this first structure, right? And those days, we're talking about 50 years ago, how were structures actually solved? They're actually built, literally built, which is called the wire model. And if any of you come to Washington, DC, and you have an opportunity to go to the National Institutes of Health, they actually have the wire model there. And this is how scientists used to measure angles and all of those things, right? Can you imagine how it was? But it was very precise. And now we don't even know what effort we have to put in to get a structure. Now, this is all the different Nobel Prizes that people have won for structural biology. And you can see pretty much almost every other year, there is a Nobel laureate who has solved the structure of something. So this just tells you how structures are very important, right? Without structures, understanding functions is hard, right? In essence, if you want a Nobel Prize, study structural biology, right? I'm not kidding. Of course, all the easy protein structures have already been solved. So all the ones that are there are all very, very difficult proteins. So any one of you interested in structural biology, I would urge you to pursue structural biology because, we find answers to these things. Of course, part of it is a joke as well, right? So why else would we study a protein structure other than, of course, winning a Nobel Prize, right? protein structure. So when you look at this structure and when you look at this structure, right, I'm going to take this off here. So when you look at this structure and when you look at this structure, do they look similar to you, to your eyes? The helices are here. Yes, of course. Yes, Betsy, it actually right. This actually looks very similar, right? The structures. But when you align the sequences, they're only 21% identical when you do an alignment of these two protein sequences. Isn't that shocking? Two proteins that have about 20% in sequence similarity or identity still can have the same structure, right? These are different enzymes, right? So what we assume, we make a lot of assumptions in science, right? So what we will say is that in order to understand functions, even when the sequence similarity is very, very low, to find the relatedness between two sequences, and if they have the same function, we just cannot just do a blast and look at them and say, oh my God, they're only 20% in sequence similarity. That means they are not related to each other, which means they probably don't do the same function. And also they don't actually have the same, they are different, but that's not what it means. So that's why in order to understand similarities between two sequences, even if their sequence identities is low, we need to have three-dimensional structures. So you can actually discover relatedness between two proteins that are not similar at sequence, but at the structure level, right? So why study protein structure? Also, although it is a lot of work and a lot of pain and suffering, it helps us to understand function, right? Without protein structures, we can understand functions precisely. Now, you all know this was again Watson and Crick. They actually got the three-dimensional structure. And of course, I have to also mention Rosalind Franklin, right? They actually found out the structure of DNA, three-dimensional structure of DNA. Just imagine if we didn't understand DNA structure today, we won't be doing any of the things we're doing today because we won't even know what pairs with what, and we wouldn't even know the stacking effects. We wouldn't even know how DNA binds to proteins. We wouldn't even know anything, right? So that was a landmark. And now these are the three people who actually won the Nobel Prize for that, physiology and medicine, right? Now again, why else would we study protein structure? We will study protein structure to understand fundamentals of cell machineries like protein translation. How are proteins made in the cell? How do proteins actually work? Do you remember any protein that is involved in the whole process of translation? Can you think of any protein that's integral to the whole mechanism of translation? Do you know where proteins are made in the cell? Any guesses? Yes, that's correct. Ribosome, my goodness. Cytoplasm, absolutely. So ribosome, correct? Many of you have said ribosome, absolutely. It's actually where it's one of the most important machinery in the cells. Are there any proteins that are part of the ribosome and the whole translation machinery? That's actually very important. What actually recognizes your mRNA? Of course, there are lots of enzymes. That's correct, Jeffrey. There are lots of enzymes, DNA polymerases and all of those things, right? So this is the three-dimensional structure of ribosome. You know, it's humongous. You know how many years it took them to actually come to this high resolution structure to look at atomic details of every single atom in the whole ribosome machinery? It took them actually 20, 30 years to actually come up with the structure. And the minute we have the structure, we can explain how the tRNA binds, what is the role of the RNA component of these ribosomes, what are all these different proteins actually doing. Exactly, we were able to explain the entire process of translation. And of course, ribosome, the biggest lesson we learned is a ribosome is a ribozyme, which means it's an enzyme, right? So there are 3 tRNA sites. We knew that tRNA played a big role, but we just didn't know where the tRNA is bound and how all of these different sites actually one went to the next and all of those things, right? So based on the three-dimensional structure, we were able to exactly locate where the tRNA binds, what are the different interactions, and all of those things. And these are the three people who actually won the Nobel Prize. Actually, he passed away, unfortunately, last year, but they're all there. So then why else would we study protein structure? We study protein structure to understand protein channels, work as they make up 30% of our cells, are membrane proteins and channels. They form all these transporters and all of those things, right? Again, Roderick McKinnon, he actually figured out exactly how things in the cell move in and out of the membrane, right? In and out. Without the three-dimensional atomic resolution and atomic positions of every single atom, we would not know exactly how these channels work and how these actually sit in a membrane, right? This is the potassium channel, and he figured out exactly what every single atom also means. You know, I run an entire course on all of this. So as you know, I'm just giving you just a glimpse of what structural biology can actually help, right? So it's a lot there. So we'll just giving you a little flavor of what structures can help, and you understand. Also, it helps us to understand enzyme mechanisms, how peptidases work. For example, have you all heard of peptidases? Do you all know what peptidases actually do? Just based on this peptide? No? Okay. So does this give you a hint? Cleaves? Yeah, it actually cleaves, but it cleaves specifically. Yes, exactly. Peptide bonds. Correct. Fantastic. You guys are amazing. So this is peptidases, means it cleaves peptide bonds, right? So any enzyme, it actually works only on 1 or 2 amino acids, right? It just cleaves something. It breaks down something, that's correct, and all of those things, right? So how did we understand exactly which bond is getting cleaved and how is it getting cleaved, which so that exactly to the details of an atomic, to the atom and to the bond, it actually helps you understand structures. And also structures helps you understand, learn about evolution. You look further back in evolutionary history and figure out how. I actually gave you an example of how 2 proteins that have very low sequence identities had the similar structures. Here I'm talking to you about 2 different protein structures, but they actually do the same function, right? So we have conservative evolution, and you know, we have different kinds where actually they come together. Of course, I don't want to confuse you because that's evolution itself is another topic for discussions. We can do it another time, right? So again, it helps us to understand that this binds to the same ligand, this also binds to the same ligand. What is common between these two are they are both dependent on PLP, right? Pyridoxal phosphates, right? And now again, D-amino acid aminotransferases and L-branch chain amino acid aminotransferases, they also have the same structures, right? So without the structure, we wouldn't actually even know that D-amino acid aminotransferase and L-branch chain amino acid aminotransferase are actually the same, which means they do the same thing. So again, it helps you understand this, right? Last but not the least, structures help design drugs. Drug design structures are integral to drug design. That is what I do for a living. This is what I do. I look at structures and I do drug designing, drug repurposing. That's actually part of my research. That's what I do for a living. So it's actually phenomenal how to come up. And I'm sure all of you have heard of remdesivir. Does it ring a bell? Have you all heard of remdesivir? It's all over the news these days. Do you, yes? Which context did you hear about remdesivir? COVID, correct? So we heard about COVID, right? So for COVID, exactly. For COVID, they said, oh my goodness, remdesivir actually works, and it actually does not cure them, but at least it minimizes the hospitalization stay for some of the patients. How does remdesivir work? How did they find remdesivir? Through structural biology. They actually docked remdesivir to RNA-dependent RNA polymerase of this COVID. And they said, well, you know, that's how they actually found the remdesivir. So they're actually using it. So there are many drugs that people are desperately trying to find for COVID, including myself. We're doing drug repurposing. How are we doing drug repurposing? We're just using all the drugs that are already out there and trying to dock it to all the different proteins and figuring out the way if it'll actually work and can we explain the mechanism, right? Now, Again, these are some more examples. So it's a imatinib. It's a tyrosine kinase inhibitor. Anytime you see IB at the end of any drug, that means it is against a kinase, right? So we have all of this. AB means antibody. So we have all of these nomenclatures. So imatinib, again, was actually the structure is available, and it was, imatinib binds to the structure. And this is, again, it's a cholesterol lowering drug, atorvastatin. I'm sure you've all heard of statins. And in fact, statin was one of the things that I put on my slide yesterday when I was talking about SNPs, right? So we have identified some SNPs that actually, some people, why statins work in some people, it does not work in some people. Why does it give adverse effects, side effects in some people? So we actually talked about this yesterday, right? So again, three-dimensional structures, right? This is again lisinopril. So this is actually, there are lots. I'm just giving you some examples of how structures were actually used in order to understand the drugs, how the drugs bind and how the drugs interact with the proteins and all of those fun stuff. I'm sure all of you have heard of ibuprofen, correct? This is something that people like me who have headaches all the time from different things actually take a lot, right? Ibuprofen. And it's a popular one, right? So again, it's for pain relieving and inflammation, relieving inflammation. So, this is how it is. So at the end of the day, many proteins are used in treatment of diseases, hormone erythropoietin. And this is, again, it's a hormone, monoclonal antibody. So I said AB, which is an antibody, monoclonal antibody. So this is an antibody molecule. So that actually binds to its, where is the pink that they're talking about? So this is the pink color here they're talking about. So the antibody binds to its antigen binding sites. What are the amino acids that are involved in this binding? All of these things. So, right. So I think I've given you enough examples of how structures are actually used in drug design. And I'm sure you've all heard of this as well, right? ACE2. I'm sure you've all heard of ACE and ACE2. Again, this has become very popular right now because of COVID again. ACE2, this is not ACE. ACE is downstream of ACE2, which is a receptor that COVID binds to, right? So these are all different examples. You know what? And last but not the least, a picture is worth 1,000 words, right? When you look at the picture, you know exactly where the ligand is binding. This is again an antigen-antibody interaction. So exactly without structures, you won't be able to understand exactly where things bind, what kind of interactions that they actually take, and all of those things, right? Again, so antigen and antibody, the way they work, I'm sure you've all learned, is by shape complementarity, right? And induced fit. So when an antigen binds to an antibody, that is actually true for any ligand protein interaction. They actually, the minute they bind, they induce some conformational changes. And so it's kind of a fit, right? So now, any questions up to this point? Any questions? So have I convinced you that structures are actually extremely important to understand functions, and they actually play a big, big role in many different things? Yes, good. If those of you there who are like, I'm not still convinced that structures are helpful, within the next few slides, I'm sure I'm going to convince you, right? Now, we know that our life is not maintained by orphans, right? We are not alone, right? Even in the cell, the proteins don't work alone. Everything is coordinated, and it's just amazing and fascinating how proteins talk to each other and how they, I call this a relay system, right? In Olympics, you have the 4x100 or relay system. One has to give the relay to the other one has to do. So pretty much it's all a network, systems network, right? It's all about networks in our cell and in our lives as well. Now, so many social networks, there's nothing that we can do without networks. So we have organs, we have tissues. Tissues are made up of cells. Cells are made up of molecules, and molecules are made up of atoms, correct? So now a cell is an organization of millions of molecules. Proper communication between these molecules is actually extremely essential to the normal functioning of the cell. And to understand communication between molecules, we have to understand the arrangement of these particular atoms. Okay, so I am seeing a question here. I would like to know, if you can actually post the question to everyone, I think that will be helpful, so that if I'm not seeing the question, then at least Deepsha or someone else will actually, who are monitoring the chat will be, so I'll be able to answer. So I would like to know if the structure complementarity can trigger a series of wrong pathway in terms of disease, like any examples. So I'm not sure if I'm understanding your question correctly, but I'm going to rephrase your question the way I'm understanding it, and you tell me if that's what you're trying to ask. So you're trying to say that something that binds, so for example, a drug, an antibody that's binding to an antigen in the cell, right? So it's because of complementarity. You're asking whether this can trigger wrong pathways. Does it mean that if another protein exists with the same shape, and that whether this drug, because of the complementarity that this may have with other proteins that look alike, whether that is actually going to cause any adverse reactions in a disease? Is that correct, Parimala? Is that the question you're asking? If that is the question you're asking, absolutely correct. That is what we are struggling with, because, for example, kinases, right? I mean, the kinases, all the kinases that belong to a very same family will have similar structures. If they have similar structures, they will have similar functions, right? So which means a drug for kinase A may bind to another kinase B that looks like kinase A, right? That's why we are having issues with cross-reactivity. We are having issues with side effects. We are having issues with non-specific binding. And that is actually a big problem that we are actually having. And yes, that's why, you know, that we are actually figuring out how to make something specific. Specificity is actually an issue, right? So we do have examples, a lot of drug. For example, Tarceva is an example that comes to my mind. It's actually, it's given for cancers. And Tarceva actually binds to a couple of proteins. Although it was actually, I don't recall exactly what it was developing, which of the kinases or which of the proteins it was developed against, the specific target, but it does bind to EGFR, it binds to several different proteins. So yes, we could have issues with that. It's a very good question. Does that answer your question? Okay, so how do you create a specificity for a drug? Jeffrey, that's a $1 million question. And in fact, I'm actually working on that very same thing. How do you become specific, right? So the specificity problem is actually is a little complicated, but yes, we can. What we can do is we usually use, if you hold on for a minute, I'm going to explain within the next coming slides. I think it'll be better if I actually show you something and then I will actually explain. It's actually a question that I'm actually working on. So hold on, Jeffrey. I'm going to answer your question in a minute. So now this is a molecular machinery, and we are going to be doing hands-on of looking at protein data bank. And so I think that we will talk about all of these things when we actually do, protein data bank hands-on. Right? In a cell, what happens is it's all molecular networks, right? So we actually have several different pathways, millions of different pathways. So what exactly happens in one pathway? If there is a problem, does it affect a pathway downstream? What do you all think? Actually, it actually does, right? So what happens here actually affects what happens here as well. That's what, again, we call it systems medicine, systems biology. It's all about a system as a whole, right? So we are all talking about all of those things. These are all the different biochemical pathways, and I'm sure that you've all actually seen all of these things. And a few different popular pathways that we all talk about is glycogen degradation, citric acid cycle, pentose pathway all of these different pathways. Why am I talking about all of these pathways? I'm talking about all of these pathways because at the end of the day, any pathway you take, right? This is the example of a citric acid cycle that's presented to you. This is a structural view of the citric acid cycle, right? So all the enzymes that are involved in the citric acid cycle, the structures are known, right? So why is that important? So if this structure this structure is important. This particular enzyme is important because the product of this particular enzyme triggers this one, right? And the substrate, this becomes a substrate of this, and the product of this becomes a substrate of the next step. That's how all these cycles work, right? So if there is a problem here, then we already know that there is going to be a problem down here. A problem here is going to impact problem here, right? So that's what happens. So until we had all of these structures, we couldn't understand exactly why it's not binding, or it's binding too tightly, the products are not being formed, and maybe a hydrogen bond is lost, and all of those things, right? So looking at everything in 3 dimensions actually gives you a more insight into the functionality of all of these things, right? So that's where it comes to high resolution structural biology. So using atomic structure to understand how biomolecules communicate and functions, right? So what tools do we use? I'm showing a lot of pretty pictures. To my eyes, they all look pretty, right? They're all pretty pictures of helices and strands and all of those things that we're going to talk about. So what do you think are the tools that we actually use to make those pretty pictures? Do you know what techniques are actually being used that actually give you those pretty 3-dimensional structures? Any ideas what methodologies are used? Yes, absolutely. X-ray, exactly. X-rays are used, X-ray crystallography used, NMR, correct? And JMOL, yes, Jeffrey, JMOL is used, but JMOL is more of visualization. So JMOL is used for looking at structures, but X-ray crystallography, nuclear magnetic resonance spectroscopy, and there is one other technique that is being used that you guys have not said. Anything else that you want to add to the plate? Yes, you're right, X-ray, NMR. One more technique that is used. So this is X-ray crystallography. This is nuclear magnetic resonance. Electron microscopy, absolutely. Cryo-electron microscopy, yes. Spectroscopy, correct. Absolutely correct. So electron microscopy, you know what? Now even a lot of structures of COVID are actually done by electron microscopy. The reason is each one actually has pros and cons of using these techniques, but of course X-ray crystallography remains as a gold standard of a methodology, and of course NMR as well, because I'm an X-ray, I'm trained in X-ray crystallography. I think this is the best, but I'm just kidding. But a majority of the structures are solved by X-ray crystallography. We have nuclear magnetic resonance and electron microscopy. Also now, because we have lots of information, remember the big data we talked about, we have lots of information enough to do modeling, computation, right? Based on what we already have from all of these different techniques. And visualization and all of those things. So these are all the different tools that we use in structural biology, right, to solve the structures. Now, this is the fun part. Yes, what are the benefits of each of these techniques? That is a whole new lecture, Jeffrey, but I'm just going to quickly tell you. X-ray crystallography, all of them actually give you the atomic resolution or the atomic positions, right? At the atomic level, they give you the coordinates of every single atom in your protein. Now, what are the advantages and disadvantages? Advantages of X-ray crystallography, it is actually, it's a gold standard technique, and it can actually, we were able to solve the whole structure of a ribosome using X-ray crystallography. You can imagine how huge a ribosome is, right? So there is no limitation on the size, right? NMR has limitations on the size of the protein that we actually can measure using NMR spectroscopy. Electron microscopy does not also have size. In fact, it's better than X-ray crystallography because you can actually look at the whole cell by electron microscopy. But what is different between electron microscopy and X-ray crystallography is the resolution of what you get out of electron microscopy is lower than what you get of X-ray crystallography. But I must say that, of course, a crystallography, there is radiation damage, and the biggest problem with X-ray crystallography is you need crystals before you have to make proteins as crystals. And I can tell you, having done that, it can be a nightmare to crystallize a protein. Not all proteins can be crystallized. So that is the bottleneck in X-ray crystallography. So each one has its advantages and disadvantages, and each one is actually complementary to the other. So NMR actually gives you hydrogen information because protons are the main center to nuclear magnetic resonance. X-ray crystallography, it's in electrons because it's X-ray beam. So electrons are the ones that diffract the X-rays. But then again, you can see hydrogens in crystallography. So we have to model it, right? So yes, you have to purify the protein and all of those things. Good, looks like you guys are very interested in the methodology. Let me see if I come up with some time to actually talk to you more about these things, right? Yes. So Jeffrey, did it answer your questions a little bit at least, right? You get a big overall picture of what the advantages and disadvantages of each of those techniques. Yes, okay. Yes, overview, good. Now structure, show and tell all, this is my favorite part of what, I want all of you to actually close your eyes for a second, just kidding, right? Don't sleep off. I just want you to imagine a protein sequence in front of your eyes, right? And just a plain old sequence, right, in front of your eyes. And now I want you to tell, if you have a structure, what can it tell that your plain old sequence, primary sequence, cannot tell you? What can structure tell you that a sequence cannot tell you? Just imagine a plain old sequence in front of your eyes, right? So what can structure tell you that the sequence cannot tell you? Just quickly, I just want to see, you guys are really, really interactive and smart. So you're able to polarity. So exactly. So when you say, Karina, polarity, you mean about charge distribution. If that is the case, yes. Fitting of molecules, that's absolutely, that is correct. Polarity, of course, charges. Fitting of molecules, absolutely. Muyiva, yes, it is correct. And Suparna, yes. Interaction, absolutely correct. Absolutely, Chandrani, I have to say. Pretty much you guys have filled my slide, right? You guys are amazing. So it actually tells you how molecules are packed. It tells you interaction, right? And I think that Karina talked about polarities in a way. I think if that's what you were referring to, it actually interaction. And then it actually tells you how folding, how does they fold, which means which part of the molecule is close to which part of the molecule, right? Just imagine, right? This helix and this helix, right? In a primary sequence, say this is residue number 30 to 35, and this is about 500 to 550. I'm just making up these numbers. From a sequence, you won't know that regions 30 to 35 is actually physically close in three-dimensional space to 500 and 550, right? 500 and 500, amino acid 500 is far away from amino acid 50 in a primary sequence. Would you all agree? But in three dimensions, because of the effect of folding, they come so close to each other, which we wouldn't know with just looking at a primary sequence. And of course, one of you actually told me about interaction of different parts. How does active sites and binding sites, Chandrani, I think, yes. So how does the ligand actually bind to, and where does it bind that are making the binding pocket? What are the regions that are exposed to the solvent? What are the regions, excuse me. And if you have two structures that are very similar, you can just superimpose them to figure out whether they're similar or not. And I think we're going to be doing this in our hands-on session when we do structures. We're going to be superimposing the molecule of hemoglobin, and we're going to a normal hemoglobin and a sickle the hemoglobin, and we're going to find the difference, right? And then this is actually electrostatic and shape. And I think one of you actually asked me about how do you make, I don't know if it was Jeffrey, how do you make something specific, right? So this is how we look at it. We actually look at charge distribution. We actually look at hydrogen bond interactions. We actually look at all of these different interactions, and we look at the energies to figure out how we can make something specific. So we may actually change one of the atoms in the drug so that it changes its whole hydrophobicity and its polarity, or I'm just using those words that you guys have used. So we can actually change the interaction between this, and I'm sure although the structures are very similar between two things, we can actually make it very specific by altering the atoms in the drug to make it more basic or more acidic, or adding more hydrogen bonds, or or introducing a salt bridge or something like that, right? So that is pretty much how, it's a lot of work and a lot of things that go in. So I'm just giving you a simplistic answer of how something can be made specific. And then, of course, with a structure, you will understand what is a quaternary structure, right? Do all structures have a quaternary structure, do you think? Do all proteins have a quaternary structure? What is your answer? No, of course, you're correct. Why? Why don't all proteins actually have a quaternary structure? So that becomes definition. You're all correct. Yes, all proteins do not have a quaternary structures. Only those proteins that come together from two different polypeptide chains, which means those two polypeptide chains actually code for two different genes, right? So when two different things come together, and quaternary structure is made, or proteins that contain two different polypeptide chains coming from two different genes. Those are the proteins that actually have a quaternary structure. And immediately you guys can give me an example of a protein that you all know, we've been talking about quite a bit in the last 2 days, is something that actually has a quaternary structure. Any guesses of a protein? Kinesins, I am not sure about that, but another protein that I actually mentioned that we're going to be, that causes sickle cell anemia. Hemoglobin actually, yes, hemoglobin actually has a quaternary structure. So we'll look at that. Then one of you actually said that we actually can do active sites and functional motifs. Yes, we actually can get active sites based on a three-dimensional structure. Okay, so now structure show and tell. Now you understand what structures can actually tell you, that you cannot get this information from just looking at just a sequence alone, right? What are the key players in this game? Proteins. Proteins play a very key role in our living system, right? They help in catalysis enzymes. They actually help in transport. There are transport proteins. They are information transfer proteins. We have hormones. So the whole protein world can be actually divided into, each one is given a job in the cell to do. And if they fail to do the job that they're given, that's when something goes wrong. When something goes wrong, and that manifests itself in, a disease of, for example, right? So everything has to be organized in the cell. Everything has to wake up at a certain time and has to let go of a certain time. If they actually don't do, then that's a problem, right? So things happen at very femtoseconds inside the cell. So things have to go on and off, on and off. It's really fun to watch things in real life under a microscope, right? So these are all different examples of classes of different proteins. We have structural proteins, contractiles, and all of those things, right? You all know these are all different examples. Each one does a unique job. And some proteins, multitask. They are given more roles to play. So which means they're very, very busy, right? Some of us have lots of hats to wear, right? So major categories: regulatory proteins, they control gene expression; receptor proteins, they bind to things. You have structural proteins. Some of them, they just have to, fold in a certain form or a way to actually understand the functions of these things. Transfer proteins are proteins that transfer something from one thing to another. Do you all know an example of a transfer protein, something that transfers something from one to another? We have a lot of different transferases in our systems. Immunoproteins, these are very, very important proteins because they are the ones that are protecting us from foreign agents, right? So your immune system is very, very important, right? And then many more, right? I'm not doing justice to everything. This is just some examples, right? This is the world. So world of folds and functions. There are lots and different, each protein folds differently. And because fold is very closely related to its function. So each protein folds differently based on the work it's given. And do you think we have a finite number of folds in this world, or you think that we have infinite number of folds or proteins? The other way to ask, membrane transport proteins. Yes, Jeffrey, that's correct. Now, do you think that proteins can fold in any way that they actually want to fold? In other words, do we have an infinite number? Yeah, microtubule bound proteins, correct. So do you think we have an infinite number of folds in this that proteins can have? The number of total folds we have, it can be 1 million, for example. No. Why? Yes, that is correct, Behu. Yes, we do have a finite number of folds. Yes, due to steric interaction. That's right. We have very finite number of folds. Do we know exactly what that number is? We still don't know the exact number, but there are numbers floating in the literature, like 10,000 folds and all of that. But my guess is that we're almost reaching the number of folds that we actually can see. Why? Because every structure that's coming out, new structure, is actually looking like something we already know, right? So the reason we have only finite number of folds is because the beauty is they all have to obey geometry. You cannot have any angle, any distance, anything that you want. You know what? At the end of the day, every protein has to follow the law of physics. So that's probably, physics is very important, and, everything follows rules. Now, do you all know that we have 21 amino acids? So why am I bringing this up? I'm sure you all know your amino acids, correct? The reason I'm bringing this up is each amino acid actually has an important role to play in a protein structure. Because each of them actually, have a different physiochemical property. So when they fold together, some of them coordinate and work well together, and others actually don't. So they group together so that they actually work well within the context of a fold, right? So each protein has a unique structure, right? And this is the amino acid sequence. And you fold this is what you get, right? So depending on where the amino acids are, they actually take sides where they want to be, right? So I'm sure that you know hydrophobic amino acids, hydrophilic amino acids, amphipathic amino acids. I just thought you all know all of that, and so I'm not actually going to be going over that. Just to tell you that hydrophobic means phobia, means fear of something, right? These proteins actually are scared of water, so most of the hydrophobic proteins are actually formed in the inner core. They actually stay away from water. Hydrophilic means these are amino acids that actually are like water, so they can interact well with water. So these are the amino acids that are actually formed on the surface of the protein. And then amphipathic means that these are amino acids that actually kind of have both properties of hydrophobic, and hydrophilic, right? So proteins actually are very, very organized. They're organized into primary structure, secondary structure, tertiary structure, and quaternary structures, right? And then protein structure has a hierarchical organization. So what is a primary structure? Do all proteins have a primary structure? What do you think? Does all proteins have to have a primary structure? Of course, yes, right? This is a no-brainer because we all start with the protein sequence, right? So everything has a sequence, and that tells you the arrangement of every single amino acids, right? So does every protein have a secondary structure? Yes, of course. Does every protein have a tertiary structure? Yes, of course, because the tertiary structure is the one that gives the fold of the protein, and that tells you how they interact with each other. Does every structure have a quaternary, every protein have a quaternary structure? We already discussed that. No, every protein does not have a quaternary structure, right? So the important things are the tertiary structures, right? How they interact. Now, I already told you, right? The sequence determines the three-dimensional structure. So everything that you want to know about a protein structure is already, the secret is already there in the primary structure, right? But the only problem is we are still facing a problem where we still don't know exactly how to get that information from ab initio using physics, first principles, right? And the next person who is going to solve this protein folding problem is where you have a sequence and you just fold it. You find a way to fold it from first principles, is the next Nobel Prize winning person in the field of structural biology. And I kind of have guesses of who that would be because I'm seeing a lot of work from that group. But that is where we're all waiting for, where you'll be able to fold a protein sequence. You just take a sequence and you give it to a machine and it's going to come up with all that. But of course, some of us will be out of a job after that, right? Because we're doing crystallography and NMR and all of those things, but first principles. But that's going to take some time because we're still trying to understand these proteins. Now, certain positions, how does a protein know how to start folding? What is the position of the amino acid that starts folding? So we have a lot of theories and hypotheses around this. Certain positions along the chain of the protein sequence actually act as a nucleation points. And chaperones, right? It's a type of a helper protein, and we know we have a lot of chaperone proteins that we have identified. And these are all the different proteins that actually help in assisting protein folding, right? They all have something that they assist in protein folding. Now, this is the primary structure, right? This is just a structure. And every protein, as you all know, has an N terminus, amino terminus, and a carboxy terminus. We always read the protein from the N terminus to the C terminus, right? That is our thing. And this is how it folds, right? So this is a primary structure. And the only thing that we actually could understand based on the primary structure is we actually may be able to say, okay, this protein could we have a disulfide branch, maybe forming a disulfide bond. But if there are 10 or 20 different cysteines, we don't know which cysteine is going to pair with which cysteine. And if the wrong cysteine pairs with the, there's a wrong pairing of these cysteines, what happens? You'll get a misfolded protein, right? So protein folding is very, very important. Now I'm going to discuss the different components. So you have basic secondary structural units of the protein. So you actually have, this is how the protein within you have hydrogen bonds, right? Hydrogen bonds are most important bonds that actually stabilize a protein structure. They actually stabilize even a water molecule, right? You have lots of hydrogen bonds. Hydrogen bonds are actually very important bonds, right? I give a whole lecture, believe me or not, on hydrogen bonds, right? Now, secondary structures is we have 3 different secondary structures, right? So you have a helix, then you have a strand, right? And then you have what is called a loop, right? That connects the different secondary structural elements. No protein can exist without, which of these 3 secondary structural elements you think that proteins cannot exist without? There are proteins that can just be helices. Yes, correct. That's correct. Loop is the one because that is a connectivity. So every single protein structure will actually have loops. But there are protein structures which may contain only helices, or there are protein structures that actually contain only beta strands. But there is no structure. Of course, there are no structures that have only loops because then there's nothing we can, they can do, right? Because they're just random, right? So they don't have a shape or form. They're so flexible. So these are the 3, and can you believe the whole world of holes is just made up of only 3 things. So life is actually simple when you look at it, but then it's still complex. But there is infinite number of ways that they can combine, right? These secondary structural elements can combine in a protein to actually give you different folds, right? Isn't that fascinating? It's just fascinating to me, right? So this is a strand, this is a helix. So in a helix, you have hydrogen bonds in consecutive amino acids within the helix. In a strand, these are different parts of the protein. So the hydrogen bonds are between 2 different strands, right? Many different strands together actually form a sheet, right? Beta sheet, right? Now these are the secondary, this is alpha helix. This is actually a beta sheet, right? So now, and that's a sheet. Now secondary structures, so they associate, these are the 3 different things. And of course the loops are actually referred to as, what do the colors represent? You mean here, the colors don't mean anything. I just color them differently so that you can see them distinctively. So in this particular, there is colors doesn't, but in this representation, usually blue is for nitrogens or the hydrophobic, and reds are actually oxygens, right? So in this particular representation, we usually use blue and reds for positive and negative, positive and negative charge distributions. We usually typically use blue and red. But in this particular case, this actually doesn't mean anything in terms of colors. Red is my favorite color. That's all it means in this particular case, right? So now they associate with different patterns of hydrogen bonds, right? All of them. And sometimes loops are actually called turns and, random coil. So loops are called different things, right? And there are lots, and believe me or not, there are lots and lots of different turns in a protein structure. And again, I do a whole lecture on that, twists and turns in protein structures, right? There are lots of different types of turns. Now, this is the tertiary structure in which how these secondary structural elements combine to form a 3-dimensional structure, and tertiary and arrangement, right? In 3 dimensions, right? So these are all in 3 dimensions. They all form together and how they interact with each other. So that is what tertiary structure means. These are all the different tertiary structures of proteins. And I must also tell you, in structural biology, it's almost like talking a new language, right? How you present your protein structure actually talks a lot. For example, this one is called a sphere. These are all spheres, right? So this is a representation of a protein structure, right? You're just representing it with spheres. Then this just tells you an overall shape. It gives you the overall charge distribution, surface representation of the protein. But here, this is all bonds, right? This is actually going to give you a wired representation that tells you every single amino acid is given to you. And so this is a representation that you would actually use for maybe showing how one amino acid residue interacts with another amino acid residue. This one is an overall tertiary structure, how the strands and where the positions of the strands are and where the helices are. So in structural biology, we use different representation to represent different things, right? To convey different things to you. Now, this is a surface representation that says blue is electrostatically positive and the red is electrostatically negative. So this is a very important representation that I use a lot to understand where a drug binds, where are the different, there are binding pockets. You can see deep down pockets. And if it is red, I should be looking for a drug that is complementary to red, right? Because like poles repel. And so we have complementarity. So this is what tells me if I have to bind a drug here, if this is a pocket, then I have to find a drug that is actually more basic, correct? So that there is charge attraction between them, right? So that's what we use all of this for. Now, quaternary structure is between different chains. And also, you know what? This tells you how two different polypeptide chains actually coming from two different genes actually come together and they form all of these different domains. And we used to call them subunits. And I stay away from using that term because it means different things, subunits. Here, there are two different polypeptides. Here in this particular case is a tetramer. This is another protein. So there are lots of proteins that actually have a quaternary structure, right? There's a whole list of them. And these are all different examples of quaternary structures comes together in a protein and functions. The majority of all the complex reactions in your body in the cell are all coming from different proteins coming together to do that particular function, right? Okay, so the reasons of secondary structure formation is the side chains, hydrophobic residues are always located in the hydrophobic core. And the main chain atoms of the same residues in most cases are also within the hydrophobic core, right? Hydrophobic is actually very important interaction, right? Because of that forms the core of every protein structure. Since the presence of the polar groups in the hydrophobic environment is very, very unfavorable to the main chain atoms, and it's also neutralized by hydrogen bonds. So most of these hydrophobic acid residues actually are inside, form the core of the protein. The two most efficient ways of hydrogen bond formation is to bring alpha helix or a beta sheet. And did you all know that Linus Pauling was the one who actually discovered alpha helix, right? Linus Pauling actually came up and actually discovered alpha helix, right? This is so-called space filling model of alpha helix. And I don't know, and he is the one that actually won 2 Nobel Prizes, right? And I am told I heard the story from someone who actually worked with Linus Pauling, David Davies, who unfortunately passed away, who was actually the first person, and I actually had the honor of meeting him and working with him actually. And he told me the story about how Linus Pauling came up with alpha helix, and I love to share that story with my students. He apparently had the flu, and he was just playing around with a piece of paper and he was folding it and all of those things, and boom, he came up. I am not sure. I actually initially thought it was a fairy tale, but apparently it's actually real. What you can actually do when you actually have fever, right? So for me, I just curl around and sleep, right? So, but that was the story behind Linus Pauling, right? It's an interesting. So the properties of various secondary structure, this is all alpha, this is a alpha helix. So as I told you, right, these hydrogen bonds are internal, and these are the hydrogen bonds that stabilize alpha helix, right? And oops. And so it goes from N terminus to C terminus, right? Alpha helix, right? And now these are the ones that stabilize our alpha helix, right? And what happens if there is a mutation within an alpha helix, right? Say that this amino acid is actually mutated and it is maybe a glycine, right? So say that this is mutated. Because of the charge distribution of an alpha helix, there is a dipole moment at the end of the alpha helix, right? That is what I want you to remember right now. We'll come back to this discussion of what will happen if there is a mutation here, right? Now, there are two different types of alpha helices, right? One is a left-handed alpha helices and one is a right-handed alpha helix. And again, you have to remember when it comes to structures, they all have to obey the rules of physics. They have to obey laws. So you cannot have anything and everything because they're not energetically favorable. So which of these helices do you think is most commonly found in protein structures? One of this is actually not found. The other, it's very, excuse me, it's very rarely found. So which one do you think is actually found? Is it a left-handed helix or a right-handed? Yes, absolutely. Actually, it is the right-handed helix that is actually formed. The left-handed helix is actually energetically not favorable. Now, does it mean that it's completely not even there? But we do see occasionally, because there are always exceptions to the rule, but it's still rare, right? Now, these are all the different hydrogen bonding patterns. So I said there is actually a charge at the end of every helix. There is a net charge because of the carboxy amino terminus, which is a positively charged nitrogen atoms, right? NH3. And here it's COOH. So every helix ends with the amino terminus and the carboxy terminus. So they actually have a dipole moment and they actually have a charge. And these distances are always the same, right? So they all have to obey laws and rules. Now, this is another representation, which is called the Seepage space filling model of a alpha helix, right? Now, there are periodic patterns that are observed. So there are proteins that actually follow patterns, right? Patterns is very important in life, right? So if you identify a pattern, we could be uncovering something about that particular protein, right? So these are all the different domains, right? So these are all the patterns. So you have all of these, you have a strand, you have a, helix, a helix strand, helix strand, helix, I'm sorry, it's helix strand, helix strand. So there is actually a pattern, right? There is a strand, there is a helix, there's a strand, there is a helix, there's a strand, there's a helix, right? So there is a pattern. Some of these patterns actually help. And if we identify these patterns, we can actually understand the functions of these proteins, right? Have you all had heard of patterns in proteins? Helix turn, helix motif, and these are all called structural motifs in a protein, right? Have you all heard of patterns? Yes. And there is a pattern. So have you all heard of helix turn, helix? And do you know what that is actually known for? A helix turn, helix pattern is known for what? Do you know? Any things that it is actually known to bind to something? Any guesses of what a helix turn helix motif in a protein structure actually binds to? Any example that comes to your mind? Helix turn helix is known for binding DNA. So how does it help you? If you find a helix turn helix motif in a protein structure, immediately you can say, this could be binding DNA, right? That's how we understand functions, right? The most common location of an alpha helix is alongside of the protein with one side of this. So you will find lots of these alpha helices on the surface of the protein. And immediately, what does it tell you about the amino acids that actually form a helix? I'm telling you that it is facing outside and the hydrophobic core of the helix will be facing inside, which means all the amino acids that actually are charged like aspartic acid, glutamic acid, lysine, arginine, that actually have a charge will be facing the outside because they can interact with water. And all the valine, alanine, all of those will be facing the inside of the protein, right? So this will actually result in a periodic pattern of all of these things. And I told you, right, there are lots of twists and turns. There are also lots of other types of helices. And I think I don't want to confuse you and I don't want to put you all to sleep right now by talking about lots of things and, overwhelming you. but I just want you to know that when you're reading a paper, for example, when you're doing your research and you read a structure paper, you may actually encounter a helix that's called 310. It just means 3 amino acids within a turn. So it's a very short stretch of helix. And then you also see a pi helix. So each one has a different definition. But these are all, are all examples. And yes, it is very likely. I'm not sure. It's actually a polyalanine, I think. It's a polyalanine, right? And so these are all, however, for a number of reasons, all of these are alpha helices, right? So these are all the different types of helices. What is the difference between, how do you define something as an alpha helix or a 310 helix or a pi helix? It all depends on the hydrogen bonding pattern and the number of amino acids that actually form them. I don't want to confuse you anymore, right? Just to say that. Now, the next secondary structural elements is strands. And I told you that strands are, you have hydrogen bonds between different parts of the molecule, right? So you have anti-parallel strands. This is the anti-parallel strand. So this is the N terminus, which means this is amino acid 1, amino acid 2, amino acid 3, 4, 5, 6, and this is 7, 8, 9, 10, right? So this is a turn, right? And the hydrogen bonding is between amino acid here and amino acid here. I forgot the number that I was counting, right? So this is the anti-parallel strand. Now the parallel strand, so this is the anti and this is the hydrogen bonding in an anti-parallel strand, right? Now in a parallel strand, so you have one, which is the thing. So this is, you have 1, 2, 3, 4, 5, 6. This is amino acid 7, 8, 9, 10. So they just parallel. And you can see that the hydrogen bond is distorted, right? Which one do you think is more commonly found in protein structures? Is it parallel strand or anti-parallel strand? Just based on what I just showed you. Because it's all, at the end of the day, it has to be stable. So which one is more common? That is correct. Anti-parallel strand is more common. Can any one of you tell me why anti-parallel strand is more common than a parallel strand? Any guesses of why anti-parallel strand is more common? Ends might have loops, that's correct, but less steric hindrance, yes. So anti-parallel, correct. Any other reasons? So I'm just going to tell you, right? So this one, right, this is anti-parallel. And so it goes in this direction, and then there is a short loop here, right? And then it goes in this direction, right? Interaction between N and C, that is absolutely correct. So you know what? So this turns here, right? So it's a short loop, and there's just a couple of amino acids, one amino acid that connects to here, that goes here, right? Now in a parallel strand, what happens is this actually has to come all the way here, which is practically impossible, right? It's practically impossible for one amino acid to be able to connect to something that's so far away. So it's energetically not possible. It's practically not even possible. So it doesn't exist. Okay, does that make sense? And this is a parallel strand, and also all these are destructive. So parallel strands that form a sheet actually are not found in protein structures, right? Because they don't, the energy, because of energetics and because of the way that these things actually form. Now that I've told you about strands and I've told you about helices, now I asked you a question, if you remember, a few slides before, and I'm going to make a statement and I want you to think about it. A mutation in a helix is extremely very, very expensive compared to a mutation in a strand, right? What do I mean by expensive? What do I mean is the effect of one mutation in a helix is really going to disrupt the protein structure far more than a mutation in a strand. Now that you've looked at both the helix and the strand, can you tell me why that would be the case, that a mutation in a helix is going to disrupt a lot more than a mutation in a strand? Think about it. I know you all know the answers. It's actually simple based on just all that I've said. Any guesses why a mutation in a helix is going to be, not that tolerable? Any guesses? Are you all tired? Looks like you're all a little bit tired. I know it's a lot, but we'll hopefully get through. Might affect interaction of protein with surrounding molecules. Just think about just within the helix itself, what happens. Alpha helix is at the surface, maybe sheets are more deeper. Dipole, yes, Marissa, you got the point. Yes, all Parimala and Suparna, what you've said is correct in a different context, but Marissa, that's correct, absolutely correct. It's because of the dipole moment, right? I talked to you about that. When you have a dipole moment, there is a charge distribution. So what happens is when you have a single mutation in a helix, right? So what happens is a mutation here actually is going to be disrupting the hydrogen bond. Where is that? This is better. Say that there's a mutation here, and it's going to be disrupting the hydrogen bonding pattern of this. But what happens is because of that, the whole effect of this is going to be felt by the entire helix and the surrounding that it interacts with. So it's going to be disrupting the entire thing. But then the same reason is, why is that not the case with a strand? It is not the case with the strand is because, you know what, if there is a mutation here, this hydrogen bond is not going to happen, but it's actually going to be compensated by the regions because it's linear, right? So that is why a mutation in a helix is far more, it is actually not tolerated because it causes more damage to the protein structure, right? A lot of these strands together as forms a sheet, right? This is an anti-parallel sheet that I told you that is actually found in protein structures. And this is the parallel, as I can tell you, like this whole thing is not even possible, right? From here, it has to get all the way here, and it's energetically not possible. It is not physically possible for a bond to be that big to connect to here, okay? Now, is it clear about all of these secondary structural elements? And sometimes there are twists in a beta sheet. A beta sheet can be twisted. And we find those things as well, right? Almost all beta sheets actually have twists, right? So that it feels really nice to look at them. The twist is always right-handed. Okay, so now any questions? I'm going to pause for a moment because I know that I threw a lot at you in the last 10-15 minutes, and I know you're all getting tired. Maybe, I don't know, depending on where you are, sleepy or hungry. So are all hydrogen bonds in a sheet in the same plane, or in a helix, they are in a different? Okay, that is actually a fantastic question. This, can you explain the dipole moment again? Okay, sure, I can explain the dipole moment here again. So what I'm going to do is get back to that slide. And I'm going to answer that question as well, Suparna, about your question. So what a dipole moment is actually, the helix goes this way, correct? So residue 1, 2, how many other residues that there are, right? Now what happens is there is a charge at this end because this is the amino terminus of the helix. This is the carboxy terminus of the helix. Where is that? So here, right? So because there is, on this top of this plane, there is actually a charge distribution along this helical plane. So there is actually a net negative charge at the end of the C terminus because carboxy, right? COO is negative, NH3 is positive. So here, this is the amino term. So there is a net positive charge at this end, and there is a net negative charge. So there is a charge distribution across the plane of this, across the dipole moment. That's what we call, that is actually along the axis of the helix that causes the dipole moment, right? Because of the charge distribution. Does that answer your question, Jeffrey? And I'm going to answer, how do you read the 3D? Is there a step-by-step? You read a 3D structure from N terminus to C terminus, pretty much. That's all we need. Now, we'll need annotation of helix. Kindly hover around it. What is, I didn't understand that question, Muiwa. And also, Suparna, did I answer your question? A peptide bond is always planar, period. Peptide bond has to be planar. So the peptide bond are always those atoms, the atoms that are in the peptide bond are actually planar, right? So that probably, hopefully answered your question, Suparna. Now, Muiwa, I don't know what what will need annotation of helix. Can you point on what constitute helix? Oh, I see. Okay, this is the helix. This whole thing is a helix, right? This whole thing is a stairway case, right? So this whole thing is it's a helix. And in a folded structure, this is the helix. Okay, does that, are you able to see my pointer? My mouse? Yes, okay, good. Okay, so did I answer all of your questions? If not, I can come back to it once we finish the lecture. And then we can have a discussion, right? About everything. So now turns are the last part. You know, we have lots and lots of twists and turns. There are many different types of turns. So you have a gamma turn, and all of them are, I'm not going to literally confuse you, but all I want to impress upon you from this is that there are lots of different types of turns, and each of these different turns has different functions. And how do you know something is a gamma turn? It is defined by the number of amino acids that form that turn, and also the hydrogen bonding pattern that forms the turn. And so gamma turn, beta turns, alpha turns, these are all the different types of turns that we find in protein structures. And there are many, many people who actually just focus their entire life on looking at these turns, right? Now, which turn is the most commonly found in protein structures? Have you heard about any one of these turns? Either when you're reading any of those things. So yes, correct. Beta turn is the most commonly found turns, right? And then we have the gamma turn and then everything else. Now beta turns, again, it is defined by the number of amino acids here and what constitutes the hydrogen bonding pattern in these things, right? So it is all depends on hydrogen bonds, which amino acid residue actually forms hydrogen bond with amino acid. If I is the first amino acid, you have 4 amino acid, I plus 1, I plus 2, I plus 3. And then you have actually I and I plus 3 distance. So it just follows some rules. And it also follows phi and psi. Have you all heard of phi and psi? Two dihedral angles that are integral to every single protein structure. And there is a plot that we plot, and I think unfortunately I may have taken off those slides just for not getting too deep and drowning into this world of structures and technical details. But have you all heard of Ramachandran Plot? Yes? No? No? Okay. So Ramachandran Plot. Okay, you've all not heard of Ramachandran Plot. Yes. Okay. So now, you know what? I think it's actually, will that be explained? Yes. Actually, you know what? I took away the slides explaining Ramachandran Plot. But you know what? When we're doing the hands-on session, I believe on Thursday on protein structures, I am going to explain to you Ramachandran Plot because I think it's important. I think I took away all of those slides to explain Ramachandran Plot. But I promise that I'll explain to you because when we actually are looking at structures and doing hands-on, I think it'll be more, clear for you what it actually means. So I definitely, and several of you have actually heard of Ramachandran Plot. It's actually a plot of phi and psi that tells you what is allowed, what is not allowed in terms of protein folds and all of those things. But I'm going to come back to that, okay? Now, in terms of turns, there are 2 types of turns, type 1 turn and type 2 turn. Oh, wow. I didn't realize I had an effect in this. I must have done it in the middle of the night. Wow. That is nice. I didn't think that there was the sound effect, but it's nice. So these are the hydrogen bonds. That is different between the 2 types of turns. In fact, if we have time today, if I finish everything I'm planning to talk to you today, then I can explain Ramachandran Plot today, okay? So this is actually in the front and this actually behind. So you're not going to have an exam in this course and all that. So, but this is just for your information. If you want to visit this later on, that there are different types of term, type 1 and type 2. What is the difference? So only specific amino acid residues are actually allowed to be in that turn. Why do you think glycine is the only possible amino acid at position 3 in a type 2 turn? What is so unique about glycine that this is the glycine that is winning the prize to be in that particular position? It's becoming important. So what do you know about glycine? Any information? Hydrogen is exactly a side chain. Other than that, what do you know about glycine in terms of its flexibility, and why is glycine always found and known to be the most flexible amino acids. Hydrogen as a side chain, that is correct. And can you expand on that, Suparna? It is actually correct. Hydrogen. What else do you all know about property of a glycine amino acid? Anything else that comes to your mind about glycine? So glycine is one of the smallest amino acids, right? So it can fit anywhere. Sometimes, you know, no steric hindrance, it's very small, because that's correct. It's actually very, very small. It's always found in, you know, most of the time glycine is found in a loop because it's so flexible, it's so small. So it can fit in many different things, right? And yeah, because it's small, it doesn't cause any. Oops, I didn't know that was coming, right? Okay, I didn't know. Sorry about that. I didn't realize that I didn't have these side effects. There may be more coming. So this is a type 1 turn, and this is a type 2 turn, right? The hydrogen bonding pattern is different, and the amino acid that this turn forms is actually very different, right? Now there are gamma turns. Just quickly, it's again another turn. There are different types of gamma turns. And again, the difference between gamma turns and beta turns is just that the hydrogen bonds and the amino acids that are involved in these turns are actually different, right? Other than that, I don't want to confuse you anymore. And I want to mention beta bulges, because beta bulges is actually amino acid that actually, there are 2 strands, and the amino acid just bulges out, right? So you actually see, why is bulges very important? Bulges are actually very, very important for protein function. Many of the antibodies actually have bulges, right? They also have bulges. Antibodies actually have bulges, right? And so bulges is actually important for functions, right? Other than that, and the other one last thing I want to talk about is proline is not found in alpha helices. Proline is actually called a helix breaker. It actually breaks the helix. It's found in turns. And why do you think that proline has taken the name and given the name of a helix breaker? Why do you think it's called a helix breaker? We don't want to see a proline in a helix. And whether we like it or not, it is still, some proteins have that. Okay, so can I go back to bulges? Yes. Do you have a question about bulges from Parimala? Yes. Okay. So bulges are something that where one amino acid is not, remember, see that this is actually, bulges are the ones that actually literally bulge out of a secondary structure, right? When you look at this, can you see that this is bulged? This strand is actually bulged a little bit, right? Because of this amino acids. Most of these amino acids are actually big amino acids. Most of these bulges are caused by tryptophans. And so it bulges out from the normal secondary structure. Does it clear your question, Parimala? And it is also very important for structure prediction and functions, right? So now, why is a helix, why is a proline called a helix breaker? What is unique about a proline amino acid that it is actually called a proline? It is an amino acid, that is correct. It is an amino acid, but other than that, imagine its structure, right? So the side chain of the proline folds back to the main chain, and so it actually causes a kink, right? It's not like other amino acids where they're actually away from the main chain atom, right? From the main chains. This is the peptide bond, right? CONH, right? So it is actually the proline amino acid turns back and connects back to the main chain, right? So it comes back to the main chain. So that's why it causes a kink. And when you see a kink in a helix, right? It breaks the continuity of the helix. It breaks the continuity of the helix. It actually also, and all of those things. Okay, so now that is it for all of the, information about all of those things. We still have major challenges in the field of structural biology. And I want all of you young minds to actually look into this, and you can actually come up with solutions to some of these problems. Yes, in a sense, it actually affects it, right? The dipole, the question is, how does the proline amino acid actually influence the dipole moment? So it doesn't actually alter the net charge that you would have because, but it does alter the polarity of its neighboring amino acid. So it affects the hydrogen bonding pattern for it to hydrogen bond with the consecutive helix interactions. It probably affects the helix interactions, right? Now, how does a sequence determine the three-dimensional? How does a three-dimensional structure relate to its function? What is function, right? So this is a question that I again ask everyone because I keep telling you, oh, the whole important goal of mission of all the scientists in the world is to understand functions, right? But what is function? What defines function, right? What is function to you all? You're sitting through this lecture, right? And I've been talking to you about structure and function and structure. And what is function? How do you define function? Everything in life has a function, right? Function to play. We all have functions to play. So what is the function of a protein? What does it actually mean? Anyone? I am sure that you all know the answer to what a function is. How do you define functions? The work a protein does, that's correct. Its intended role, that's correct. What it does for the cell, fantastic. All great things, right? So biomolecular activities, correct. What does it do? So all of each one of you have actually defined function, right? So what does it do? What does the protein do? And what are the important things? Protein sequence, structure, and structure to function, right? So that's pretty much what it is. So I'm telling you structure and it helps understand functions, right? The enormous variety of protein functions, right, is based on their high specificity for the molecules that they interact with, right? It can be interacting, and these are interacting partners I call as ligands, right? A protein interacts with another protein. In this case, it's a protein ligand. A protein interacts with the DNA. That is, a DNA is a ligand. A protein interacts with a small molecule. Like majority of the proteins in the cell actually bind to small ligands like ATP, GTP, GMP, and all of those things, right? Which you've all heard of. These are all called ligands. A protein's function actually is defined by what it actually does, right? Some proteins cleave majority of the proteins are actually enzymes, which means they do, it's a catalytic function that they have. They cleave something, they transfer something, they actually reduce something, oxidoreductases, or they actually, each one has different functions, right? Enzymes, but not all proteins are enzymes, right? And also those things, right? So it all depends. But do you think they say that a protein actually has 1,000 amino acid residues? And do you think that all the 1,000 amino acid residues are actually important for that protein to carry out its function? Right? This has always puzzled me, right? In nature, we have like enzymes, right? Say that an enzyme is like 1,000 amino acid residues, right? But is 1,000 amino acid involved in its catalytic activity of actually cleaving something and doing this electrophilic attack or electrostatic attack or not? No, actually, then what is the role of the whole thing? So how can you break the 1,000 amino acids into smaller bits, right? So you're right? So all the 1,000 amino acids are not important for that enzyme. It's only 1 or 2 amino acids, which we call the catalytic amino acids, right? Histidines, aspartic acid, catalytic triad. Maybe there are 3 amino acids that are important. But then what is the function of rest of the other amino acids in that? So each amino acid or each part of this actually has a function, right? So that is again a function, right? So you can break up these big proteins into smaller bits. And how can you break a big protein into smaller bits? What are those called? You can break a protein into, well, yes, at the end of the day, it is all, points down to each amino acid is from a codon. That's correct, Suparna. So we actually break the whole protein into domains, right? Domains are the functional units of a protein, right? A protein can have 10 different domains. A protein can have just one domain. A protein can have like, 2 domains, all of those things. At the end of the day, it's all about domains. But can you break down the domains into smaller bits? Can you break down a domain? Yes, motifs, correct. You can break down the domain into motifs, correct, right? So hence protein ligand interactions is central to molecular recognition. So anything and everything, it's all about interactions, right? So hence it is no surprise that the biological function of a protein are so connected to its three-dimensional structure, and even a little damage to these structures is often a reason for loss of dramatic effect that it has. Majority of proteins, one single mutation, and we talked about Megan, yes, subunits. That's correct. So you can talk about 1,000 amino acid residues. We can break that up into domains. Then the domains, we can break them into different subunits within the protein. Then we can break each one of them into motifs. And then of course, Jeffrey, we can get back to the codon level at the end of the day. If we keep breaking them into smaller bits, finally we'll figure out which amino acid is coded by which one. At the end of the day, we can come down to all of those different levels, correct? So even a little damage to these structures is often the reason for loss of dramatic, right? So we discussed a lot about monogenic diseases where one single mutation is actually causing a lot of damage to the protein sequence. Then it's actually a base pair. We can start with the codon, base change in the codon, either a base pair, and then that codes for a different protein because of the mutation and variation, and then because it is structured to function, right? One mutation affects it, right? So that is where. So again, this is what we talked about yesterday, correct? So we have diseases, we have inheritance, and we have all of these genes that are possible, right? So all of these things, single mutations, right? Now we're going to talk about sickle cell anemia, right? So sickle cell anemia is, do you know what makes the color of your blood red? What protein is involved in making the color of your heme? Yes, correct. All of you are very correct, right? So hemoglobin. Hemoglobin is the one that makes the color red. And what else makes it red? So I'm sure you've all seen all of your veins, right? And have you all, I'm sure, of course, iron, that's correct. Iron in that, absolutely correct. Iron in that. But what happens to this, right? I'm not sure, of course, you're all too young, and but I'm pretty sure that you've gone to a doctor's office and maybe you've had a blood drawn, right? At some points in your life, maybe you've had blood tests just to check everything is okay. Oxygenate, right? That's absolutely correct, Jeffrey. So I'm sure that all of you have gone to a doctor's office and they draw your blood, correct, in a little tube. And have you all noticed what happens to the blood in the little tube as they're collecting the blood? Is it always red? Or what happens to that? Have you all noticed blood when they start doing a blood draw on you and they actually have a tube? And yes, it darkens, correct. It darkens, right? It is, I would say it's more purple, blue, correct? So you've all seen that. And even your veins, right? When you look at your hands, you see a lot of this bluish thing, right? Greenish thing, right? It changes. That's because, you know what, there's no oxygen, right? It's taken away from your body. So it changes color because oxygen. So oxygen is actually extremely important for our life, right? It also, so hemoglobin actually binds to oxygen. So now, excuse me. So hemoglobin is actually a very, very important protein in your body. It carries oxygen from the lungs to all the tissues. You need oxygen. Every single cell breathes. For breathing, you need oxygen. So it carries all this oxygen. And it carries carbon dioxide from the tissues back to the lungs for purification, correct? And the normal levels, each of us actually have a normal level of what a hemoglobin should have. And I'm sure some of you have heard, oh my God, you're very anemic, which means you don't have enough hemoglobin. So they put you on iron, you need to have, your iron content is low, and all of those things. So hemoglobin is a very, very integral molecule, correct? Now, It consists of 4 different polypeptide chains, right? 2 alpha chains and 2 beta chains. So do you think that the alpha chains and beta chains actually come from the same gene? Yes or no? No, correct. They actually come from 2 different genes, right? And so that's why we talk about 2 polypeptide chains. And did you know that the definition of a gene actually has changed in the last 10 years? When I went to school, it was the definition of a gene was a gene is something that codes for a protein, right? That is a definition that I was taught in school. But now I'm sure all of you know that the definition of a gene has changed, because a gene is something that codes for a polypeptide, because there are proteins that are pre-existing, like in the pre-form and pre and pro forms, and different chains come together to make a protein and all of those things. So you know what? The definition has actually changed, right? All this data is changing our minds about understanding different things, right? So now you have, it all has a heme group, porphyrin ring, and each chain contains a heme group that carries the oxygen. And of course, it contains an iron atom in the center that actually binds to the oxygen. And a hemoglobin molecule carries 4 heme groups and carries 4 molecules of oxygen, right? It carries it all over the place. Now, so this is the heme, it's planar, and oxygen and deoxygen bound form and not form, and a complex of protoporphyrin 9, and it's a ferrous iron 2+ is present in the center of the heme. It binds to 4 nitrogen atoms of the ring, right? And additional bonds with histidine residues of globin chain, and it actually binds to oxygen too, right? So all of this you actually know, right? Now hemoglobin is a great molecular machine. So what drives most of the protein interactions, right? So hemoglobin, I'm telling you, binds to oxygen, right? So it has to bind. But think about it. Does hemoglobin always stays in an oxygen bound form all the time? Does hemoglobin does it stay bound to oxygen all the time? No, of course not. Now, you know what? There is a molecule called KRAS. I'm not sure whether Kirstine, it's involved in pancreatic cancer. It drops it off around the body. That's correct. So what happens is every protein is given a function, and it has to actually do the function. And so it has an active state and an inactive state, right? So every protein in the cell actually goes through active state, inactive state, active state, inactive state. So when it does something, it binds exactly. So it actually has to switch its roles, right? And now hemoglobin may have other things to do as well, right? And each protein actually has different functions to play. So it cannot keep doing the same thing over and over again, right? So what happens is it binds, it doesn't bind. It binds, and so I call it the active, inactive. So there is actually a hemostasis in the body where every protein binds active state, and it has to get to inactive state for the next cycle. So it's a cycle, right? So when something doesn't let go of something, that's when you actually get diseases. In majority of the diseases, some of the proteins always, or the genes actually always stay in their activated form. For example, in pancreatic cancer, there is a protein called KRAS, or of course, KRAS is a protein that actually a gene that is implicated in many different kinds of cancers, but it is actually very integral to pancreatic cancer. What happens is it binds to GTP, right? A signal comes, it binds to GTP. You all know GTP, right? Guanine triphosphate, right? Guanosine triphosphate. So GTP, it binds to GTP, and it's an active state. When it binds to HTP, then something else comes and hydrolyzes that GTP to GDP. So it's a triphosphate, then it becomes a diphosphate from GTP to GDP. So when it becomes GDP, when GTP is hydrolyzed to GDP, then it becomes inactive. So KRAS in the cell always is an active, inactive, active, inactive. So it is from GTP to GDP, GTP to GDP. But what happens in cancers? It always stays in the GTP form. So it's always activated. So then it actually is always, so everything that KRAS, all the downstream and upstream proteins and genes that are dependent on KRAS are all gone crazy because they're waiting for a signal from KRAS, but this KRAS is not getting hydrolyzed. So what happens? It's always in an active form. So what happens is always in an active form, that's what is a problem, right? We cannot even design a drug. You know why? Because the GTP is binding there and we cannot even bind anything else there, right? So that is the problem. So same thing, that hemoglobin, yeah, more active than GTP. I'm not sure, Moiva, if I'm understanding. So you mean GTP is more, yeah, exactly. GDP, you mean, more active than GDP. GDP is the inactive state, right? So now what happens is all of them actually go through a lot of changes. So this hemoglobin molecule, just look at this. Look at how, so this is the oxygen, right? So look at what changes happen between the oxy state and the deoxy state. It keeps the whole, these domain and this domain actually change with respect to each other, right? They keep changing with respect to each other, right? So they go through huge changes with respect to each other, right? So that is what happens. A lot of conformational changes actually happens. And then there are different types of hemoglobin, right? So this is normal hemoglobin, and then this is fetal hemoglobin. When a baby is there, we have hemoglobin F, it's a fetal hemoglobin. And oops, sorry. And then we have a hemoglobin A1C, which is hemoglobin A when it's glycosylated, which means sugars are added to the hemoglobin and the levels are very high. And I'm sure that you've heard of A1C that is being measured in people with diabetic, right? Who are diabetic, that you are measuring A1C levels, right? Then we have abnormal levels of carboxy hemoglobin and all of these things. So there are different types of hemoglobin. And what happens when the hemoglobin gets into trouble, which means it gets to a mutation, right? So the genes for the protein chains of hemoglobin, they show small differences within different human populations, right? So actually, you know what, between you and me, there are slight changes, but not the function. Of course, it retains the function, but there are slight changes between you and me. There are some amino acids that may be different and differences within human population, right? And amino acid sequence of hemoglobin slightly different from person to person. And in most cases, the changes does not affect the protein function and are often not even noticed, right? We talked about SNPs and we talked about a lot of different things that actually can be different from different things and all of those things. And in most cases, actually, they do not affect all of us, right? And we won't even notice that there are changes in hemoglobin, right? And in some cases, these different amino acids lead to major structural changes. So one such example of a major change, which you all know, is actually sickle cell anemia, where there is one mutation from a glutamic acid to a valine. And do you know why? Now that you know the properties of some of these amino acids, you know all these structural elements and all of those things. So why do you think one single mutation has actually caused such a major change in a structure, and hence it's causing a disease, right? So say, for example, I'm going to ask you this question. Say that instead of a glutamic acid, say that it mutated to an aspartic acid. Do you think you will still see the same dramatic change in the protein structure? Why is changing a glutamic acid to a valine so, you know, it's causing so much of problems? Any ideas? The hydrophobicity. Okay, so you mean that valine is more hydrophobic than glutamic acid? And then, no, because both are acidic. Actually, valine is actually a hydrophobic amino acid, Shupana. It's actually not an acidic acid. And then it affects the helix. Yes, it affects the helix. I actually told you, right? It does affect the helix. That is actually correct. Binding of oxygen site is, of course, we already know that changed, but I'm asking why glutamic acid to valine is so pronounced. Just focus on those two amino acids and the properties of just those two amino acids. Oh, yes. Okay, good. Super. Yeah. Thanks for the clarification. Yeah, you're absolutely correct. You're absolutely correct. So tell me, just focus on aspartic acid, glutamic acid and valine. Let's look at the properties of these. Glutamic acid is actually a negatively charged amino acid, right? Its pKa is around 4.3 or something like that. But a valine on the other side is actually a very hydrophobic amino acid. So you are actually disrupting the complete charge distribution by just this one amino acid from a charged to an uncharged. So it's a huge change. And I think, as Moyuga said, I think it is affecting the of the distribution of the helix, or the three-dimensional positioning of the helices. So what happened is this change in one region of the protein has affected a lot of different regions in the other parts of the protein, right? And so, yes, in majority of the diseases, it is just one amino acid that can actually disrupt based on what that a particular amino acid is, right? So now this is a phylogenetic tree of all the different families of hemoglobin that we know, right? So you can actually see there are so many different families of hemoglobin in different species and different things, right? But of course, having said that, all of them are conserved in their function. All of them are conserved in the region that actually does the oxygen binding and all of those things. If you do that, but these are small changes in other parts of hemoglobin. There are different changes. So you can actually see that there are so many different, families of all of these things. Now, you all know about sickle cell anemia, right? One such example that where there's one single, you already know, glutamate, right? So it's actually changing in one base change. And then it is giving from normal protein, it is actually giving a sickled protein. What is happening is the structure of hemoglobin is actually changing. It becomes sickled. The mutation allows the deoxygenated form of hemoglobin to actually stick to each other, and it produces stiff fibers of hemoglobin inside the blood, right? So you know what? So it causes, it's not able to bind to oxygen. There is not enough oxygen. The red blood cells are sickled like this, right? From this shape, it all becomes this shape, right? So isn't that amazing, right? So all of this, as it's going, it becomes sickled. And this in turn, what happens is it deforms the red blood cells, which normally is a very smooth disc shape, and it becomes the sickled shape, right? And these distorted cells are fragile and often rupture, leading to the loss of hemoglobin, right? So what happens to loss of hemoglobin? You become anemic, right? And anemia is actually a serious disease. It's actually fatal. Anemia is very fatal. You can die from being anemic, right? So that's why it's very serious. And interestingly, a lot of this sickle cell anemia is very prevalent in countries like Asia and Africa and all of those places, hot places. It's very thing, right? But then, you know what? In diseases, we've noticed there are more than a couple of examples, right? So can we compare the fibers to I am not sure I understand your question, Suparna, when you are talking about actin and its relevance to sickle cell. Can you elaborate? I just don't know. Hold on that question, okay? We'll just open up for questioning, and then we can actually, all of those things. Information out there that RBCs are sickled as protected. Yeah, exactly. So Muiwa, you stole my thunder right now. That is exactly what I wanted to talk about, right? So majority of some of these mutations, you know what happens is it causes one disease, but then it is protecting you from another disease. So it is almost like we have to pick and choose which disease we want, right? So people with sickle cell anemia, they're protected by, they won't get malaria, right? Again, there are so many examples where people with one disease are actually, they're protected. So I think that people with, I can think of, there are several examples, right? It was just in my head right now. So there are several examples of how one can protect you against another disease, right? I think even people with the receptor for Ebola, that's I'm blanking, actually are protected against Niemann-Pick disease. People with Niemann-Pick disease actually are protected from getting Ebola, right? So you know what, but there are lots of, and then there are people with some polymorphisms are protected against inflammation, right? So some of these, you know, actually cause one disease, but then they're protecting you against another disease. So I'm not sure if that's a good thing or a bad thing. You know, I don't know which one you would pick. There is a cure for malaria, so I just don't know how you think about this. So you know what, and what does it mean? And the same gene is implicated in multiple diseases, right? So we are going to be talking more about those things as we're doing the hands-on. So I think I'm almost at the end. So all I want to convince you today, and the take-home message for you today, is structure defines function. Have I actually convinced you that structure is important? And have I actually convinced you that structure? Yes, thank you, Betsy. Actually, I am, yes, fantastic. And structure is very important for function. Without structures, we can have a life, you know what I mean? And so now, essentially, good, thank you very much. Now I'm going to, the next 5 to 7 minutes that we have, I'm going to answer some of your questions, and then maybe I'll walk you through what to expect for tomorrow and what you can be prepared for tomorrow. Okay, so I'm going to get back to one of the questions that I actually did not answer. Okay, Suparna. So, okay, so what was the question? I think okay. Since hemoglobin fibers deform the RBCs in sickle cell anemia, can we compare this to the way that actin fibers affect the shape of a normal? Yes, when you're talking about the interaction that happens in a fiber, right? So the patterns of interaction that happens in a fiber, even Alzheimer's, right? You actually have beta amyloid fibers. So when you talk about fibers, we are just talking about oligomerization of these protein 100-fold, right? That's pretty much what we're talking about, right? So yeah, in terms of comparison, you actually can compare, yeah, because fibers are more, what do you call it, gives the shape and all of those things. So in that sense, absolutely correct. You can compare them if that's what you're trying to do, yes. If I'm isolating a new protein and want to know the structure, so okay, so it's a long haul. So if you want to isolate a protein, the steps involved in from protein to a structure is, you know, many different steps are involved. First, you have to purify your protein. When I say purify your protein, that means you have to purify your protein 200%, which means when you run a gel of your protein, it is just a single band. Not only that, it should have only one isoelectric species. So you need a highly super pure protein for crystallography, right? Once you purify the protein, you have to find conditions to set up crystallization of this protein. So you have to crystallize this protein. And if you are lucky, if you have a green thumb, then once you have crystals, then you take these crystals and you have to find an X-ray machine to put these crystals in an X-ray diffractor machine. It's a huge machine. And then you collect data, and then from that data, it is like a long haul. There are so many different steps that are involved, from taking a protein, from purifying to a three-dimensional structure, right? If you're interested, I can meet with you one-on-one to walk you over the detailed steps, because it's a lot to explain right now. But I'm happy to give you, there are like 5-6 major steps involved in taking a protein from sequence to structure. Yes. If the purity is not great, I would not even attempt to crystallize, because it's not going to work. If the protein is not pure, don't even attempt. Nothing is going to come. We're going to be a waste of your time. Right? Okay. So now, any other questions? Is there any concept that I talked about that just went over your head that I can, talk, spend a minute or two? If there are any questions that I can answer, please let me know. I'm sure you're all tired and exhausted. I'm just so excited with this group, very interactive group, amazing group. And I'm very thankful for that. So if I'm isolating and you want to, Bulges be sites for, can Bulges be sites for drug binding? Yes. Yeah, sure, why not? But it is a case by case basis. So it's not like a blanket statement that I can say that bulges are, every bulge you can target to find something. It all depends on the context of that particular protein. So you can explore that, because bulges come up with very, usually most of them are actually on the interfaces. So we do look at them. Any example to study? A lot of antibody-antigen interactions actually have bulges. Antibodies as structures that I actually have solved in my life for my PhD actually had a bulge. So when we see bulges, we make a big deal out of that. Yes, I've seen bulges in my own. So antibodies are, because antibodies have a higher content of tryptophans than any other protein. So we see a lot of bulges in antibody loops, hypervariable loops of antibodies. So I don't think, I'm not sure, there is no need actually Suparna for having the bulges for protein structure, but we just see them because, for example, if there is a bulky amino acid residue for some reason is in the middle of a strand, it disrupts the hydrogen bonding pattern. So what happens is it's almost like an extra amino acid in that particular strand, right? So there is no hydrogen bonding pattern for it. So what happens is it ends up just kinking and bulging based on the foci angles that it actually has. That's all. Other than that, there is nothing more to these bulges, right? There are lots of review articles on these bulges, but it's not that the protein requires bulges. It's just the other way around, that the evolution nature has created them like that, right? And in some cases, at least in my case of the structure that I solved, I found a functional consequence of that particular bulge in way it interacted with the peptide antigen that I was actually studying, right? So it made a very strong salt bridge with the antigen that I was studying. So it's a case-by-case basis, right? Okay, gene was it codes? What's the, so a gene is actually something that codes for a polypeptide. That's what we define a gene right now, that it codes for a polypeptide instead of a protein. Okay, so okay, so now let me go and see if I've not answered any other question. Can you please explain the need for bulges? Okay, I explained that. The need, okay, did I miss anything? You said parallel strands don't exist in proteins. Well, yes, exactly. That's correct, Megan. Parallel strands do not extend. As I told you, it's not energetically favorable to have parallel strands in the sense of that. But there may be short stretches in a protein that could be parallel. So I would say that it's an exception to the rule rather than the norm. And so the norm is anti-parallel strands. Okay, so any other questions do you all have? Of course, I'm here with you for, another week or so. So we can see if you have any other questions, you can actually, bring it up in the next couple of days. And you can review this and if something comes up, right? So possible state, will that be explained? I'm not sure, Parimala, which graph you're talking about. What is a pure protein? A pure protein, Marissa, means that you just see one band on your gel. That means it's just one species, one form of species in your entire suspension. For designing a research project to analyze a protein, what should we look for to make sure it actually works? So when you're talking about, Jeffrey, if you're talking about for designing a research project to analyze a protein, for your research project, if you're talking about, picking up an interesting protein for research, I would pick to see whether the protein actually has a three-dimensional structure, because then you can do a lot more with it. Whether the structure actually has a bound ligand, that is even more better because you can analyze ligand interactions. Then you can actually look at structures that are implicated in a disease. And then it's even more interesting to figure out why that, why that protein is implicated in that disease, and even more if there are some polymorphisms that have been found to be correlated with that particular disease. So this is a rule of thumb that I tell my students. for my course for them to do their projects. I tell them pick a structure that has a ligand bound, pick a protein that's implicated in a disease, pick a protein where there's a lot of literature that is known so that you can actually, look at things and, at least the biochemistry of it is worked out, right? Now, Parimala, are you talking about something that I showed today? Or I don't even remember, but we can get back to it tomorrow. So you just tell me whether I talked about it today. Did I show a graph today? I am not sure, but let me, you can tell me whether it was today. Was it today? Along with Ramachandran Plot. Okay. Okay, I will look into it and I'll get back to you tomorrow because we are running out of time today, but can you remind me tomorrow if you're going to be there tomorrow? You can remind me tomorrow. And I will, because I may end up forgetting, but then you can actually tell me tomorrow, you bring it up and then we will address that. I will look to see if the slides actually had any graph and I can explain that to you tomorrow. Yes, I guess that's it from me. Deepsha, do you have anything?
No, I don't have anything to add. I just wanted to walk everyone through once more to the organization page before we go. I think that will be helpful. Okay, so I can share my screen. And I just wanted you all to just see the organization page and where you can find the different resources. So this is the Genomics in a Virtual Lab organization page. Now you see in this forum section, we have posted all the different topics that will be covered or you will have a problem with. So under this topic, session recordings and video tutorials, you will see that the video of the first session has been posted. So if you don't see that email which I've already sent to you, you can also refer to this section right here. So I will also post today's recording here. And you can continue to see the different resources. When you go through the courses, if you have any problems with the pipelines, you can post it around here. So I'll be sharing this link again. And also we keep on posting about top performers of the week. So that is something that I want you all to see and update your profiles. So what does an updated profile look like?
Sorry, Vipcha, to interrupt now. We have another meeting, so I have to start that meeting. So it's on my Zoom. So I think we are also exceeded time. So if you can just go back the next time.
Yeah. Thank you, Mohit. Like you can go and
thank you. Thank you so much for the wonderful lecture. It was like very intriguing and learned so many things. Thank you so much.
Yeah, thank you.
Yeah.
Okay, guys, have a good day. Good night and good.
Thank you, everyone. Please update your profiles. Okay. Thank you so much.
Bye, everyone.
Bye bye. Thank you, Dr. Sona.
Yeah, thank you. Bye bye.